obviously for the history of one of the most unlikely scientific pioneers of our age, the laboratory mouse.
This is the story of the most unlikely scientific pioneers of our age. Heroes whose achievements have changed the path of humanity. They turned the chance discovery of penicillin into a drug that saved millions of lives. Thanks to them, the dream of transplant surgery is now a reality. And because of them, one day we may even be free from childhood disease. Yet for nearly a century, their contribution to medicine has been largely overlooked. Who are these heroes of modern science? They are laboratory mice.
They're extremely clever, agile animals. I have enormous respect for them.
They can serve as basically surrogate humans for studying human conditions.
Most of the knowledge we have about human biomedicine comes from laboratory mice.
This is the tale of man's real best friend. The rise of the mouse to scientific stardom began with a rare breed of human, the mouse fancier. To many of us, these little creatures are common vermin.
These nice.
But to them, the mouse is an object of deep fascination.
When I was working, you used to have a stressful job, and you come home and you go out into the shed, you pick a mouse up and stroke it, and all the cares of the world seem to disappear.
Today is the highlight of the Mouse Fancier's calendar, the South of England show. Derek Creasley, a tarmac technician from Kent, has a passion not just for keeping mice, but for breeding ever more exotic varieties.
They're very interesting little animals, they're friendly, and I like following the genetics of them as well sometimes.
This mouse is one Derek has crossbred from 2 different varieties and named the Himalayan Satin. But the judges are quick to point out its defects.
Unfortunately, this mouse hasn't quite got what we're looking for. Its ears are a bit light, and as you can see, it's got a little chip out of its ear as well.
But Derek's mouse is such a special breed that despite its flaws, it walks away with a prize.
Oh, he hasn't done too brilliantly. He won his class, but only because he was the only entrant.
But for the fanciers, breeding the perfect mouse is satisfaction enough.
You have the same sense of achieving something when you do achieve a top honour as even somebody who breeds racehorses to win the Derby or something like that.
Except we don't win £300,000.
No. So there's no way of getting rich out of it.
Yeah, we're not retiring, not yet anyway.
This occasion may seem quaint, but it's only thanks to the dedication of a breeder like Derek that the mouse has come to play such an important role in some of the biggest breakthroughs in modern science. It all began with one remarkable woman. 100 years ago in New England lived Abby Lathrop. She was a poultry farmer, but not a very successful one. She failed miserably at raising chickens and was forced to start a new line of business. Abby had long been a dedicated mouse fancier, so she decided to turn her passion into a living and breed mice.
Abby's original hopes were to sell to the mouse fanciers of the Massachusetts area. She thought it would be a rather small side business. She would sell 200 or 300 mice to these people, and then they wouldn't need her because they would breed their own mice with the pairs that she sent.
But when her mice started breeding, something strange happened. Abby noticed that some of her mice developed tumours. And there was a pattern. The tumours ran in families.
She noticed that some of the mice had more tumors than others. She noticed that some of the mice were getting particular tumors in particular places, and so was concerned about, what kind of things are breast cancers, what kind of things are skin cancers. And so she was very observant in that regard.
Abby was fascinated and brought her families of tumor-prone mice to the attention of scientists from Boston's Harvard University. They too were intrigued. If cancer was inherited in mice, perhaps the same could be true in humans. Were the mice telling them something? This was the start of the controversial link between medical research and the laboratory mouse. Now there are 25 million mice in labs around the world, most of them descendants of Abby Lathrop's original mice.
Well, we owe an enormous debt of gratitude to her. She was the mother of all fanciers, if you like, and her stocks have moved literally all over the world.
Clarence Little was among those scientists who learnt of Abby's work. He was a 25-year-old PhD student. But to start with, his interest was not mice.
Little came to Harvard wanting to study dogs, dog genetics in particular, because he came from a long line of dog fanciers. And so the story goes, one day his mentor skidded a live mouse at him across a benchtop and said, "Learn everything you can about this." And Little became hooked, and there's plenty of science that he was hooked. His zoology notebooks show him doodling mice in the margins, very realistic mice for that matter. So he was definitely the mouse man of Harvard.
Clarence Little was intrigued by Abby's observation of mice with a hereditary tendency to develop cancer. He was a pioneer of the then infant science of genetics and was working on a radical new idea that cancer in humans might be caused by faults in merely a dozen or so specific genes.
Little believed that cancer had a hereditary part to it. Being a man who could visualize the future, he saw that you could not do this with humans. You had to have some way in which you could get animals that were all alike.
Little decided to use Abby's mice as the basis for his research. So he and his team got all the mice together in their Harvard lab and spent long hours studying them intensely. With unfortunate consequences.
When the mouse researchers would travel home from work in the afternoon after having spent a whole day at the mouse house in rooms filled with ammonia-soaked shavings, they would stink, and they noticed that the citizens of Boston were not too happy about their presence on the subway and would turn up their noses at them. So they decided in the end that what they would do is just stay at the mouse house and live with the mice.
So Little's work was kicking up quite a stink. Eventually, he did the decent thing. He and his mice moved nearly 300 miles to the wilds of Maine to set up the Jackson Laboratory. There he could continue his groundbreaking work and smell just as he liked. His first task was to breed mice which were virtually genetically identical. That way, any cancer-causing mutations would really stand out. Researcher Skippy Lane worked with Little and knew the importance he placed on being able to breed a suitable mouse.
In the mouse, we can breed brother-sister till we get to a point where we have animals that are exactly alike one another. And we can grow them in large quantities. This is the way in which the genetic background of the animal becomes identical in all these animals, and whatever experimental work you then do with them will have the same response.
Little interbred several strains of mice for 20 generations, at which point each strain became virtually genetically identical. One was to become the scientific star of the future, the C57 Black 6.
Well, the black 6 mouse has been around the longest of almost any mouse strain, and it's been used by more scientists for more experiments all over the world than any other single strain. And so there's been a huge body of scientific information accumulated that is important in understanding human health issues, based just on the C57 black 6 strain.
The black 6 has turned out to be the mouse of choice for many scientists, for a whole host of reasons.
They have some rather interesting characteristics. They're not particularly aggressive mouse. They've been used in studies in addiction, for instance. They're gluttons for alcohol and other drugs.
But it could all have been very different. Because Clarence Little and his mice were about to discover the risks of living in the sticks. On the 23rd of October 1947, a forest fire swept through the area.
It was a disaster, a total disaster.
It tore through the libraries and laboratories at Jackson.
The fire was probably the most devastating thing that could ever have happened.
Most of Little's specially bred mice, including the black 6, were incinerated. Only a handful survived.
Several of the researchers I interviewed talked about going on site the next day and having the smell of burned mice permanently imprinted on their mind. They said they would never forget it. The significance of the 1947 fire for the research community was that it wiped out the largest stock of inbred mammals in North America and possibly the world.
Yet Little's mice had taught him something before they died. They had shown his cancer theory was wrong. The few genes he'd been concentrating on were not the cause of inherited cancers. The role of the laboratory mouse in medicine had been secured. Around the world, mice were beginning to transform medicine. In 1940, Britain was fighting the war against Nazi terror. Thousands of servicemen were dying, not of their wounds, but from the terrible bacterial infections which came with them. Tiny microorganisms were eating away at the tissues of those infected, and the doctors were virtually powerless. The tragedy was that many of these deaths were unnecessary. Back in 1928, Alexander Fleming had already discovered the answer: a mold which seemed to kill many bacteria. But initially, even he wasn't terribly excited about it.
He didn't see a clinical future for it. He thought it might be very difficult to extract any active principle. So he simply wrote a paper saying he had observed this mold, which he called penicillin, which was clearly inhibiting killing off bacteria.
It would take 12 years and a new team from Oxford University to realise the significance of Fleming's mould. Norman Heatley and Howard Florey wondered whether penicillin in a purified form could become the basis for a drug that might cure the bacterial infections. But they felt they couldn't just inject it directly into patients. They needed another way to test it out.
It would have been amoral to try out an quite unknown drug in a very impure form on a human being if you had not already tried it on an animal. That would have been quite unacceptable.
So they turned to the mouse, and for 3 reasons. There were plenty of them. They were seen as expendable. And critically, they had an immune system very similar to humans. At 11 o'clock on the morning of May the 25th, 1940, the Oxford scientists injected 8 white mice with deadly streptococci bacteria. The 4 so-called control mice were given nothing else, but the remaining 4 were then also injected with penicillin. Dr. Heatley's diary records what happened next.
I stayed at the lab till 3:45 a.m., by which time all 4 control animals were dead. May the 26th, Sunday, left the labs at 3:45 a.m. when it was already beginning to grow light.
The future of penicillin lay with the fate of the remaining mice.
Went to the lab at 11:45 and found all 4 treated mice alive and well. It really looks as if penicillin may be of practical importance. Had a discussion till 1:15 with the professor, who wanted me immediately to produce 100 litres of penicillin broth per week.
The penicillin had worked.
My husband was a very diffident person, and there was no eureka moment in his diary, but he must, I think, have felt very eager as he set off again for the lab that Sunday. And he subsequently noted that he had, in dressing, he had put his pants on back to front, and that was the only observation he allowed on his own state of mind.
Only as a result of the success of these mouse trials could tests begin on humans. Within 2 years, penicillin was in full use. The first of a whole range of drugs we now know as antibiotics.
Over 50 years, this medicine saved a just incalculable number of lives. It really completely revolutionized treatment.
Antibiotics remain the towering achievement of 20th century medicine. And so much of that success was made possible because of work with the humble mouse. But it didn't stop there. The war was producing a whole host of urgent medical challenges. And once again, the mouse would be called into action because of its similar immune system to humans. Peter Medawar was one of the giants of medicine at that time. He was a pioneer of skin grafts, treating airmen terribly burnt when shot down. But grafting a patient's own skin wasn't always possible. So Medawar had tried something else, grafting skin from a donor. But again and again, he hit the same problem. The recipient's immune system rejected the donor graft. To get around this, he needed to know how immune rejection worked. So he created a special mouse and tried to fool its immune system. First, he injected cells from a white mouse into the young pup of a brown mouse, because he believed a young immune system may accept foreign cells as its own. The results were groundbreaking. When the brown mouse grew up, it accepted a white skin graft with no signs of rejection. It was a discovery that has become the basis of all modern transplant surgery.
The significance of his work has been that it's helped us to understand really how the immune system works at the most basic level. And only if we can understand that can we start to learn in more sophisticated ways and in more different ways how the immune system actually does function.
The humble mouse has contributed to saving the lives of thousands of transplant patients. And even today is pushing the frontiers of our knowledge into complex areas of immunology. Geraldine Williams was struck down with rheumatoid arthritis at just 28. For 22 years, she has lived with the disease.
Impossible task.
During that time, the pain has spread across her body. It means even the simplest tasks are a trial.
It hurts fingers, it hurts your back as you're bending forward, it hurts your hips, hurts your knees. Complete nuisance. Just impossible. It makes you think, don't bother with the socks, is what it makes you think. It's painful, it's time consuming, and it's extremely frustrating just to get a sock on.
Rheumatoid arthritis is an autoimmune disease where the immune system attacks the body.
Painful, slow, and sore.
As the disease has progressed, Geraldine and her husband Dave have been forced to adapt their lives.
It's been totally life-changing because, I mean, without doubt, that the plans that you start off with as a young married couple and having children have to slightly alter as you progress through life with the arthritis. As it got slightly worse, and maybe the hands or the feet, we had to drop that thing and then maybe find another thing to do, which suited a way of life.
It'll be jigsaws next.
Tommy knows.
We're good at Tommy.
Yes.
Rheumatoid arthritis is a really nasty condition. It's by far the worst form of arthritis. It doesn't just rot away joints and make people really ill and run down. It actually can shorten your life. And if somebody develops rheumatoid arthritis and it's not treated in a timely and appropriate manner, a life expectancy can be cut short to the same degree as if somebody had developed coronary artery disease or some forms of cancer. So rheumatoid arthritis can be a killer.
In a lab in America, this mouse has been specially bred to develop rheumatoid arthritis. Studying the animal's immune system has helped researchers understand, at a cellular level, what causes the body to attack itself in this disease. And this has led to new classes of therapeutic drugs being developed. Despite her moral reservations, Geraldine is grateful these animal experiments have been carried out.
I'm an animal lover, but practically I have to say that it has to be done. There doesn't seem to be many alternatives. They can't do it on humans, so it has to be animals, I'm afraid. Selfishly, I want to be better. It's as simple as that. I'm afraid I have to go along with it.
Today, Geraldine is meeting Professor Moots, who will select a new treatment for her.
Hello, Mrs. Williams.
Hello.
Which joints are particularly sore at the moment? Your shoulders. Can you reach for the sky?
No.
How high can you go?
I can do sort of to that.
Right.
That's
and
my
that's
limit.
About your highest?
Yeah.
Geraldine is given the first dose of her selected treatment, one of a whole new class of drugs. This one works by disabling the very cells responsible for attacking her body.
So we're looking out for any redness of the site.
Even on this drug, though, Geraldine has only a 40% chance of making a full recovery.
Okay,
fine.
Even so, back home, Geraldine and Dave are excited.
Look what I found.
In a fortnight, they plan to visit their two sons who live in Australia. It will be the holiday of a lifetime, and Geraldine has a dream. But in her current condition, the dream seems remote.
To be able to go up to Sydney Bridge would be quite something. At the present second, I don't have any chance of doing it. In however many weeks it is, after this medicine, I expect to do it.
It will just be like when they got to the top of Everest. It'll be exactly the same feeling.
Should I take a flag?
Fulfillment, ambition.
And a flag.
Mm-hmm.
It's 3 weeks after Geraldine's first dose and half a world away. Sydney Harbour Bridge is twice the height of Big Ben. The climb takes 4 hours and is a challenge for even the most able-bodied.
This is where we hope to be walking up tomorrow.
Only if the drug that Geraldine has been taking is working well can she hope to make the climb.
Okay, we're on. So this is the start of the bridge climb. This is the building we're starting from. Here we are, all geared up, ready.
Geraldine prepares herself. Her family goes ahead. And Geraldine is right behind them. On the way to achieving her dream.
Top of the world, Pa!
Okay, top of the world, Pa.
They are. You, not a bit out of breath. Yahoo! Wonderful. Wonderful.
Here we are.
Mice have played a considerable role in making recovery from diseases like rheumatoid arthritis possible. And it's all because they have a similar immune system to ours. But it turns out mice and men have far more in common than that. It's Maine, it's the Jackson Laboratory, and it's Mutant Mouse Day. An open day for scientists on the lookout for the perfect rodent research partner. What's remarkable about these mice is that they have naturally and spontaneously developed the same genetic conditions that are found in humans.
Have you got a Huntington's here?
Muriel Davison has spent the last 35 years spotting them.
I chose to go into this field of finding new spontaneous mutations because I wanted to do basic research that would enable me to help human beings, obviously. And the best way I thought to do it was to find all these spontaneous mutations and make them available to other scientists to do research on. So, I mean, it's kind of like my research has expanded 100-fold on every mutant when other scientists work with it. It's like discovery every day. It's like being a pioneer. What's important about these mice is that they have a condition called muscular dystrophy, and it's also a condition that happens in human beings. It's a naturally occurring change in the DNA, so it's just an accident that happens when the DNA is being remade, and it happens the same way in human beings. So they make a model for the same condition in human beings.
This animal has the mouse equivalent of Usher's syndrome, a disease of the inner ear in humans.
It circles, and that's because its inner ear, which is responsible for balance, just like in people, has this degenerative tissue, and it doesn't have normal balance anymore. And a mouse that has that problem will circle.
This mouse, named the stargazer, can't help but jerk its head skywards because it suffers from a type of epilepsy. The fact that mice and humans share not just similar genes, but similar naturally occurring genetic defects, has raised hopes that mice like these may point us towards cures for some of the scourges of our age. And one black 6 mouse did develop a small mutation that was to have a big impact.
The research assistant who was working with that strain thought this was a pregnant female, and she set it out. Well, the next week she was just as fat as she was the week she set out. The next week she was just as fat as she was the week she was set out. In fact, the animal remained fat and never produced any offspring. So this was the first obese mouse, which looked very much like a tennis ball with a tail on it.
The obese mouse fascinated geneticist Jeffrey Friedman. Had this mouse become fat because of a genetic mutation? And if it had, could human obesity be caused by a similar mutation? Friedman's work revealed that the mouse lacked one critical gene, the gene that produces a hormone named leptin, which regulates the hypothalamus, or appetite center in our brain. Without the hormone, the mouse could not tell that it was full, so it carried on eating. It seemed that all you needed to do to cure obesity in mice was to inject leptin.
One of the people I was working with was injecting it into animals in the lab, and he came back to me and he said, "You got to see this." And what you saw was these fat mice who normally hardly move at all, running around the cage like a normal mouse. And actually it was the fact that their activity had changed was the first thing that Jeff noticed, because of course it took a little more time for the weight to come off. And at that moment we really had a pretty good idea now that this was an element of a new hormone system that was going to play a key role in regulating weight. And that was just an absolutely exhilarating experience that, I mean, it's just indescribable.
Here was an apparently simple way to cure millions of obese humans and make billions of dollars. News of the miracle hormone spread. A Californian drugs company paid $20 million for the patent, and their shares went through the roof. Word spread across New York, and to Keith Grubert. He had weighed almost 20 stone for most of his adult life.
I tried many diets, but in the end, I'd gain the weight, I'd lose the weight, I'd gain the weight, and in the end, it would just go back to being what it was. No weight loss.
To Keith, leptin promised instant salvation.
I was exuberant. I felt that, maybe this would be the final thing that could actually work and burn the fat away and reduce my fat cells and, get me in the kind of shape I was hoping I could actually get back into from my high school and college days.
Keith started his course of leptin and waited for the pounds to fall off. But nothing happened.
I guess my feeling of disappointment and letdown was that it wasn't just a matter of just taking a pill. I had to inject myself twice daily and keep all these capsules and keep all these needles. And it was very difficult to deal with because of the fact that I put myself out there and it just didn't work. And I felt that the promises that they had put out for it, was the biggest letdown of all.
10 years later, Keith still weighs nearly 20 stone. So what went wrong? Scientists now know that unlike obese mice, most obese people produce their own leptin. But their conscious desire to eat can override its action.
Every living organism has to regulate its nutrition, and there's in every organism, including humans, there's a sort of a hardwired network that represents the basic just drive to eat, which is indispensable for survival. And we can study that system perfectly well in mice, in fact, brilliantly well in mice. In humans, however, there's something on top of that, which is our cortex, oftentimes opposing what the basic drive is telling our brain.
So the fat mouse has taught us a valuable lesson. For some people, there is a genetic component to obesity, but most human conditions are down to a combination of several complex factors. And there it might have ended, until advances in genetic technology promised to open up an entirely new set of possibilities. The new millennium heralded one of science's greatest advances. The Human Genome Project sequenced every gene in the human body, but the lesser-known Mouse Genome Project was also set to make a splash. And crucially, for the very first time, scientists had the detailed information to begin to examine, one by one, the effect each mouse gene had on the animal. Now, scientists did not have to wait for naturally occurring mutations. They could create designer mutant mice to order. So Alan Bradley and his team at the Sanger Institute in Cambridge set about creating a new breed of mouse called the knockout mouse. Each one a tiny bit different, each with just a single specific gene either altered or eliminated. To do this, Bradley took mouse DNA and manipulated one solitary gene. The altered DNA was then inserted into a mouse embryo, and the embryo planted into a host mother. When the mouse was born, it would carry DNA with the single gene difference.
The scientific community has generated about 3,000 different knockout mouse strains, so that's about 10% or so of the total number of genes in the genome. So there's probably another 25,000 yet to go if we just do it once. And of course we don't just want to look at each gene once, we probably want to look at each knockout in a variety of ways. So we might take out this little bit of the gene or this end of the gene, and so each gene will be looked at probably in several different knockout mice, and so the numbers are going to be probably of the order of 100,000 by the time we're really totally done with this task.
By discovering the effect each altered gene has on the mouse, scientists hope to learn the effect of the equivalent gene on humans.
That has allowed us to understand the function of many thousands of genes and also make many human disease models, because we can, by manipulating genes in mice, understand the consequences in mice and therefore hopefully develop therapies for humans who have similar kind of genetic changes.
The hope is Bradley's knockout mice could hold the key to cures for thousands of genetic disorders. But super mice are expensive to develop and vulnerable to disease, so they and others like them require considerable care. This is Harwell Laboratories' brand new £20 million mouse house. Researchers studying the mice here have to take extreme precautions to safeguard the animals' health.
We go through air showers. We have multiple change of clothing, special shoes. We have another change of clothing to enter the actual ward itself. We then change our gloves again. We use alcohol for holding our gloves between cages. So as many things as we possibly can put in to dilute anything that is any possibility of us actually bringing something in which will come in contact with the animals.
This is Harwell's inner sanctum, the British Laboratory Mouse Breeding Centre. There are 13,000 cages here with up to 3 mice per cage. That is, unless...
No. And so I've put them in, and they've now been buried within the sawdust, and it'll give the animals something extra to do within the cage.
So the mice here are encouraged to indulge some of their natural instincts. There's sterilised paper for building nests. This may look like a toilet roll, but in fact it's a rodent tunnel. But these mice are protected for good reason. The hope is they may hold the key to curing further human disorders. Not just insights into how our genes affect our bodies, but insights into how our genes affect the most complex of all human organs, the brain.
Because of the ability to manipulate the genes in mice, we've been able to understand how genes, and the very particular genes, are important in behaviours. This has been wonderfully useful for understanding diseases, where there is a gene that is mutated in a human, and we can study how it works in the mouse. But we can also go the other way around and say, why don't we study the very molecular mechanisms in the mouse brain, understand them first, and then use that knowledge to explore how the human brain works. This has been a very successful strategy.
Dr. Pat Nolan is in charge of the neurobehavioural unit at Harwell. He's investigating the genetic link between sleep patterns and psychiatric disorders in humans, with the help of a genetically modified mouse and infrared binoculars.
We find in humans that there are disturbances in the body clock associated with many psychiatric disorders. One example would be seasonal affective disorder. And we also know that there are disturbances in aging and in many neurological disorders associated with body clock disturbances.
What Nolan needs to find out is which particular genes affect sleep patterns and how.
We normally expect for a normal animal to see about 30,000 revolutions per 24-hour period. And this roughly would be converted into about 6 kilometers. The majority of this activity takes place at night time.
It's when the lights come on that Nolan can observe how the sleep patterns of the mouse have been affected by its altered genetics. As a nocturnal animal, this mouse should now go to sleep. But it doesn't. Nolan now believes he may be on the way to uncovering crucial sleep genes.
The sorts of experiments that have been so productive in terms of finding genes that we can name and detailed study, that is an incredibly difficult thing to do in humans, and in fact the rate of such discovery of genes in humans has been very, very low. Using the mouse, we've been able to study and identify them perhaps 10 times more efficiently.
The hope is the mice at Harwell may be able to shed light on the genetic basis of other complex behaviours difficult to study in humans, like addiction. Scientists have long believed the likelihood of becoming an alcoholic may be down in some part to the interaction of several genes. But identifying which genes play which role has proved immensely difficult.
This mouse carries a mutation that causes it to have a preference for alcohol. In normal terms, mice do not like to drink alcohol. This mouse has a preference for alcohol, and it drinks probably 4 times more than you would expect it to drink. Now, in human terms, this would be the equivalent of drinking 2 bottles of whiskey a day.
Dr. Nolan gives his mice a choice of alcohol or water and measures the amount of each they drink in 24 hours. By finding the gene that makes the mouse more prone to drink alcohol, scientists hope to develop a test for alcoholism genes in humans.
Addiction is complicated in that multiple genes can play a role, and in humans it's therefore difficult to identify what these components are. We have a simple situation here where we have one gene affected, and we do not find out everything about alcoholism, but we can identify some components that are abnormal, some genetic components.
Experiments like Nolan's are beginning to shed light on the genetic components of complex behaviours. But as with many animal experiments, there is a price.
The cost of this experiment is basically that we need to generate animals that carry this mutation. As you can see, however, the behavior of the animals, apart from the fact that they drink more alcohol, seems to be pretty normal. The benefit in terms of humans is that we may establish some of the factors that are critical in the development of alcoholism.
Other less controversial laboratory methods may be suitable for some research. But many scientists believe mouse studies are the only way to investigate certain behaviours.
There's a lot of things you can do in cultured neurons and on computers, and as part of our research program, we are doing a great deal of that, in fact. But there's one thing that cells in dishes and computers don't do. They don't show behavior. They don't show the emergent properties of the brain, which is behavior. If you want to explore how an animal can learn and understand its environment, you have to study the animal. A cell does not learn and explore and understand its environment.
This may be controversial research, but scientists believe work with mice is beginning to contribute to invaluable knowledge into how our genes affect our brain development and even our learning.
Oh, that was close.
Leah Wilkinson lives in New South Wales, Australia. While she has no learning difficulties, some male members of her family are not so lucky.
I grew up with my cousin and my uncles, and we all had fun together as a family, and it wasn't until I actually became older and started to realise there may be some differences with the male children in our family. And it wasn't until I actually started to compare to the outside world that I really realised there was a problem.
Underarm not over.
Come on, get it!
My great uncle, who's since passed away, he was the eldest, and then there's my uncle, Uncle Graham, and my cousin Michael. They had difficulty understanding some what I thought were simple concepts.
We're doing fine, are we? Yeah,
Michael has done much to overcome his learning difficulties. 5 years ago, he got together with Leonie, who suffered brain damage in a car accident. She helps him get through life.
He has a hard time understanding problems. Quite several times I have to help him with spelling things, even.
Sometimes, like...
Explaining what words mean.
Because if I look at a word and I'm not sure what it means, I ask her, what does it mean? She can explain the best she can. Things like that.
On a day-to-day basis, the family doesn't let it get in the way of normal life.
How much do you think we'll need?
Maybe all the sausages.
Okay.
But its repercussions are hard to ignore, because Leah knows the learning difficulties in her family are no coincidence. Some female members of her family are carriers of a mutant gene that affect 1 in 2 of their male children.
Plate as well, or we'll just take them out the way they are? Take them out. Because we might not need those, I'm not sure.
But when a geneticist first told her of the problem, there was no way of knowing who carried the mystery gene.
She was very supportive and basically said, well, at this point in time, we'll take people's blood. And she was able to say what she could actually offer at that point in time, which 17 years ago wasn't a whole lot.
Not knowing if she carried the gene, Leah made a life-changing decision not to have children.
I wasn't prepared to have a child with a disability, an intellectual disability, and so, yeah, I guess being of childbearing age and in my early 20s, thinking, well, if I do want to go on and have children, it's going to be a very stressful event. So I think I opted to wait and just perhaps have a child later on if I felt more emotionally able to deal with those issues.
Lucy Raymond, a consultant in human genetics at Cambridge University, is studying the genetic basis of learning difficulty.
We've looked at groups of humans, and we have been able to identify a particular gene that carries an abnormality, and it turns out that this is in one of the molecules that the mouse people have already begun to do detailed work on. So it looks like some of the models of learning and memory that the mouse genetics people have highlighted and identified may be highly relevant to human learning and memory.
This is the DLG3 mouse, a mouse with a tiny mutation on just one part of one gene, a mouse with learning difficulties. And at a cost of £50,000, it's also a very expensive mouse. Scientists at the Sanger Institute are using the DLG3's sense of smell to test its capacity for learning.
Now we're about to look at the ability of a mouse to learn, to recognize and remember. And you can see here a normal mouse sniffing a cotton bud that has mouse urine on it. You can see it's very interested in it. It's spending a lot of time looking and smelling it. It's recognizing it. It's learning all about it. It's wondering where it's come from.
But that's only half the test. Just 5 minutes later, the procedure is repeated. Has the mouse remembered what it has just smelled?
So now we can present the same odor to the normal mouse, and it's immediately smelling it, it's recognizing it, and moreover, it can tell that it is the same smell that it's had before, and it recognised that very quickly, which is why it's spending less time looking at it. In other words, this mouse has learnt something, and it's also showing the ability to remember it.
Now it's the turn of the DLG3 mouse.
What we're about to see is a mouse that carries a single gene mutation. Now, this odour is presented on the cotton bud as before, and this mouse isn't showing as much curiosity initially about this odour, but you can see it spends some time having a quick smell and trying to recognise this odour. And now the same odour is being presented to the mouse carrying the mutation a second time, and you can see it spends a little bit of time smelling with it, but just as much time as before. It's not showing any evidence that it can remember having smelt this odour before. What we've seen is a very clear difference between the normal mice and the mice carrying the single gene mutation. In the normal mice, they showed the ability to recognise, they showed the interest in learning, and they showed the ability to learn. Moreover, they showed the ability to remember what they'd learned. But the mice carrying the mutation, on the other hand, they weren't particularly interested in learning, and when they tried to learn, they didn't seem to be able to remember what it is that they had learnt. These mice clearly have a rather serious learning disability.
The work on the genes which help mice learn has contributed to an important finding for Leah Wilkinson and her family. The DLG3 gene that caused the mouse to have trouble learning is the same one responsible for the learning difficulties in Leah's family. With the mutant gene identified, Leah could at last be screened to find out if she was a carrier.
When the results had come back from the genetic testing, when the genetic counsellor actually said that I was unaffected, it was like a huge weight had been lifted off my shoulders. I was just, after waiting 17 years for an answer, and I didn't realise how much it had actually affected me until I actually had the answer. And then I realised that it had impacted on my life significantly, and yeah, it was a very emotional moment for me. And I was just so happy to tell my sisters as well that, you know, I found out that Mum wasn't affected, and that means we aren't either, and we can go on and have normal male children, and I was very excited.
The result changed Leah's life. She and her husband are now trying for a family.
We're at the tip of the iceberg. We're beginning to find genes where there's a very clear causation between a gene abnormality and a problem. I think there are very many more genes to be found. After all, the brain is a very complex organ. And so I think there's plenty of work to be done.
In Britain last year, there were over 2 million experiments on mice. Across the world, mutant mice have helped scientists push the boundaries of medical research. In America, these glowing mice have had a gene from a luminescent jellyfish inserted when they were embryos. It helps scientists trace the effect of mutated genes in the body, and the hope is it could lead to a whole new range of genetic therapies. In Boston, this now infamous mouse with the unusual ear is in fact a tentative first step towards growing organs for human transplant. But now a very different type of mouse may help us gain an even greater prize: the power to live longer and stave off aging. At the Jackson Labs, where the first great mouse experiments took place, Dr. David Harrison has spent the last 30 years trying to find the genes that make us grow old. But the standard laboratory mouse could only take him so far. To unlock the ultimate mystery of aging, he had to look somewhere else.
You have to remember that the standard mouse strains all came from domesticated mice. And when you domesticate, that means that you have actually bred, you have very severely selected for animals that produce offspring very rapidly and that grow very rapidly. And those characteristics don't go with long lifespans. In the wild, on the other hand, there are environments in which the animals have their best chance of passing on their genes if they have smaller litters over a longer period of time. And those animals develop sets of genes that cause them to age more slowly.
So Harrison turned to a truly wild mouse, Mucus molossinus. Molossinus had one remarkable quality. It seemed to have a gene that slowed down aging and increased its lifespan by 10% compared to other species. But finding the gene responsible proved immensely difficult. By 2003, Dr. Harrison had managed to narrow its location down to one specific chromosome. But he needed to go even further.
And what we're looking for is genetic markers that are associated with increase in lifespan. So we can make the piece of the chromosome that increases lifespan smaller and smaller and smaller, ultimately leading to trying to find the mechanism that causes this increase in lifespan.
Honing in on the individual gene for aging is a painstaking process. But recently, Harrison has made a significant step forward.
From the wild molossinus mice, a small region of chromosome 8 seems to be associated with increase in lifespan, and an increase in maximum lifespan that in the mouse is roughly equivalent to 10 years in a human being.
Harrison believes that once he locates the specific gene he's looking for in this wild mouse, scientists will know where to begin to look for their equivalents in humans. It's a quest with a distinctly personal edge.
Working on aging, I have at least the vision that when my time comes and I meet death, I can look him in the eye and I can say, one of these days, some of us are going to end you, or at least retard the hell out of you.
The tiny mouse may yet be the key to unlocking the mighty mystery of life and death. Unlikely though it may seem, the astute observation of an extraordinary mouse fancier a century ago has led to a revolution in healthcare and has secured the mouse's place in history as a hero of modern medicine.
Cause, effect and current understanding of lightning. Nature Strikes Back on Monday at 9:30. But stay with us tonight for a look at the attempts of scientists over the centuries to discover how bats can possibly fly, defying all known laws of aeronautics. Superbat next, here on BBC 4.
30 years ago, acclaimed director Peter Hall and a rural Suffolk community made movie history.
I thought that if we could use real people in real circumstances, I thought it might be able to capture this second.
I can't put plumstone in my mouth and talk for anybody. I am just what I am.
What was strange was how people who were not related became a family.
Return to the village where it all began in Aikenfield Revisited, followed by the original film tomorrow from 9 on BBC 4.